Hi everyone. So on behalf of the ESDR board, welcome to this new edition of the ESDR Kitchen webinars. So I am Julien Seneschal, a dermatologist in Bordeaux, and today is a recipe book where new techniques for research dermatology are presented. So now I give the word to my co-chair, Professor Enikö Sonkoly, to introduce our today guest speaker, Professor Lars Norlen, for this recipe book. Please.
Thank you, Julien. Good afternoon, and you are all welcome to this episode of the ESDR Kitchen. And it is my great pleasure to introduce today's speaker, Associate Professor Lars Norlen from Karolinska Institute and from Karolinska University Hospital, where he also works as a dermatologist, a senior consultant. Lars Norlen has also been a colleague of mine in Sweden, so I know him very well. And it is really a pleasure that he can present at this ESDR Kitchen today. So what he's going to talk about today is the cryo-electron microscopy of the skin barrier, and this is something that he has been studying for more than 30 years. I think that cryo-electron microscopy is something that rings a bell for many of you because the method received the Nobel Prize in 2017. So we are all curious to hear how this method can be applied for skin and especially for the skin barrier. So please, Lars, you're welcome to share your screen.
So thank you very much, Enikö, for the introduction and for inviting me. So today I will talk about the skin's barrier, a cryo-electron microscopy based overview of its architecture and stepwise formation. And I will start to talk about the methodology, cryo-electron microscopy of vitreous sections that we have worked with for over 20 years. And it started with that I worked with Professor Jacques Dubochet, who is the inventor of cryo-electron microscopy of vitreous section and who got the Nobel Prize in 2017 for his developments. So we worked at the end of the 1990s and beginning of 2000s with the development of the cryo-electron microscopy method. And then in 2005, I went back to Karolinska Institute, and there we continued to refine the method, especially for work on skin. So basically, what is cryo-electron microscopy of vitreous sections, and what differs from conventional electron microscopy? Basically, it's an electron microscope, but the samples are native. So there is no sample preparation whatsoever. So the samples, for example, the skin, are just frozen by throwing them into liquid nitrogen and immobilized. And this means that the specimens are preserved in their native state down to the atomic level. So it's a very, very simple method. One takes a sample, a piece of skin, for example, from the arm or from the abdomen. You put it into liquid nitrogen to vitrify it, and then you cut it into ultra-thin sections, which are then posed on cool-down electron microscopy grids, which are inserted into a cryo sample holder, which then is mounted in a cryo-electron microscope, and then you take the cryo-electron micrographs. And what should be noted here is that in cryo-electron micrographs, since there is no preparation, the image or the contrast is only derived from the bioatoms themselves, nothing else. There is no staining, there is no preparation. So in reality, a cryo-electron micrograph represents an atomic density map of the specimen. So this is what it could look like. This is an image of the horny layer. It's just because it's the whole skin that is frozen, but here in the image you only see the horny layer. So this is the first horny layer cell, and this is the 20th horny layer cell. So here is the surface. So you have all the layers of the horny layer of human skin, and the cells, the horny layer cells, are these elongated structures here, and the dark bands, this is the extracellular space where the skin's barrier structure, the membrane structure that resides in the stratum corneum extracellular space, is placed. So this image, what you see, all the contrast here is directly derived from the bioatoms themselves, nothing else. So there is no staining, no preparation. So usually in practice, we take a medium resolution image to start with, to orient ourselves. So here is at a slightly higher resolution, the stratum corneum cells with the extracellular space here. And then we choose an area where we want to do really high resolution electron microscopy. And in cryo-electron microscopy, since there is no preparation, there is no or very, very little background, which means that the resolution is much, much higher than in conventional electron microscopy. So usually a resolution around a few angstroms can be retained. That is pseudo-atomic resolution. So in this case, we've chosen to focus on the extracellular space, the dark band. That is where we have the lipid matrix, the skin's barrier structure. And usually when we take the really high resolution images, we do not take one image, but a series of images, usually 3 images in a row at exactly the same spot. And we take them at different focus levels in the microscope. So we take 3 different images, 1, 2, 3, at 3 different focuses. We only change the focus in the microscope between the images. So you can see the images are quite different, and they represent 3 different optical representations of the same underlying structure. So we get a lot more information by getting images at different focuses of the microscope. So now the question is, what is the underlying structure here? You see the lamellar structure, that is the extracellular space, the skin's barrier structure. So what is the underlying molecular organization? And what we do to determine that is that we make atomic models. So we use information we have about the system that we are interested in. And in this case, it's the skin's barrier system. And there we know, for example, the composition. We know that it's composed of lipids, and we know that the lipids are ceramides, cholesterol, and free fatty acids in a roughly 1:1:1 molar ratio. Then we use that to build an atomic model. That is done, we do a simulation on that model. That is, we simulate in the computer what would that model look like in our electron microscope if it were the truth. So we get simulated images of the model that we have built, and then when we've gotten the simulated images, we compare the simulated images with the original cryo-electron microscopy images from skin. And if there is any difference between the original and the simulated, we make a new model, change a few atoms, and do the whole process again. And usually during a structure determination, we make several thousand models until we arrive at a model that has optimal correspondence between simulated and original cryo-electron microscopy data. So basically, the process is like this. So here we have the molecules that we know constitute the skin's barrier. It's ceramides, free fatty acids, and cholesterol in a 1:1:1 ratio. We have some water, and we also know that about 15% of the ceramides are acyl ceramides. That is a ceramide with linoleic acid ester bound to the top of the long chain. So now when we have the composition, we make a guess. That is, we build a model that we think might be correct. And in this case, we built this model. And now we want to equilibrate the model. So what we do is that we calculate the interaction between every atom in every molecule and all the other atoms in the system. So you see here, the molecules are composed of atoms. Every little sphere is a single atom. So the red atoms, these are oxygen atoms. The white atoms, these are hydrogen atoms. The green atoms, these are carbon atoms in ceramides, while the orange atoms are carbon atoms in free fatty acids, and the yellow atoms are carbon atoms in cholesterol. The blue ones are the carbon atoms in the acylceramides. So for every single atom, let's say this hydrogen atom, we calculate its interaction with all the other actions in the system, and which you can understand is an enormous calculation, and then it's redone every few picoseconds until the whole system is equilibrated. That is, it has found its optimal packing. Basically, it's a self-assembly. So it's done by molecular dynamic simulation. So we start to run it by molecular dynamic simulation until it has found its minimum packing organization. And the differences could be very, very big. The molecules could completely change form or change place with each other. In reality, what you see here is this is the molecular dynamics model, but it's repeated indefinitely in all three directions. So the real model looks like this, infinitely in all directions. And then we take this atomic model and insert it in our virtual electron microscope. That is our electron microscope, but in the computer. And then an electron microscopy simulation is done on this model, and we get an output of simulated electron microscopy images that we choose to simulate at focus levels of the microscope that corresponds to the ones that we used in real life when we took the real cryo-electron microscopy images in skin. So basically how this, so here you get the simulated electron micrographs at different focus levels of the microscope. So basically electron microscopy simulation works like this. We have the complete electron microscope in the computer. So first the electron source is simulated, and then all the optical steps, all the lenses in the electron microscope are simulated. And finally, the interaction between the electrons and the detector. So basically it works like this in the simulation process. First, the electron source is simulated. Then the model, the atomic model, is inserted into the virtual electron microscope, and the interaction between the atomic model, the specimen, and the simulated electrons is simulated. Then all the optics of the microscope are simulated, and finally the detector interaction of the scattered electrons is simulated, and we get an output of simulated electron micrographs. That is what this atomic model would look like in our electron microscope. And finally, we compare them with the original cryo-electron micrographs. That is these background images here, and these here, these are the simulated images from the model that we have chosen. And if there is any difference, the model is redone. The whole process is redone from scratch. And usually we do a few thousand model simulations before we arrive at an optimized model. So now we've used this technology to analyze the architecture of the skin's barrier structure at the molecular level, and also all the formation steps that is visible in cryo-electron microscopy. So here is an image of the full skin, and we will focus on the epidermis, and especially on this zone between the living viable epidermis and the dead, the horny layer, because it's here the skin's barrier is formed. So if we enlarge that, we see here the topmost stratum granulosum cell. Here is the first stratum corneum cell, the second stratum corneum cell, the third, fourth, and fifth stratum corneum cells. And what we see in the cryo-electron microscope is that in the topmost stratum granulosum, we have the lamellar body system, which also is called the tubular reticular network. And basically it's composed of interconnected lamellar bodies. The lipids of the skin barrier, they are formed in the transgolgi, and then they are transported out into the extracellular space between the last stratum granulosum cell and the first stratum corneum cell. And what we see in the cryo-electron microscope is that the lamellar bodies is more or less exclusively are composed of a multigranular, a fine granular structure in the native state in cryo-electron microscopy. And then the closer we come to the interface between the stratum corneum and the stratum granulosum, small patches of lamellar structures appear. That's what's marked with blue here. And these are lamellar structures with a periodicity of 50 to 55 angstroms. That is a periodicity of 5 to 5.5 nanometer. If we move up one step to the extracellular space between the first and second stratum corneum cell, we don't see this lamellar 50-55 angstrom pattern, the blue pattern here anymore, but we see a different pattern, which is brown here, which is more fine lamellar and with a periodicity which is much smaller. It's of 20 to 25 angstrom only, so 2 to 2.5 nanometers. If we move another step up to the intercellular space between the second and third stratum corneum cell, we see yet another pattern. So this fine lamella has disappeared, and now instead we have a fine lamellar pattern which is asymmetric and with a periodicity of 55 to 60 angstrom. And then finally, above the third stratum corneum cell in the extracellular space, we have this lamellar pattern, which is marked yellow here, which has a periodicity of 110 to 120 angstrom, that is 11 to 12 nanometers. And this fills out the complete extracellular space of the remaining horny layer. So this is the major skin barrier structure that is found everywhere in the major stratum corneum and represents the main skin barrier in our skin. So now I will start to show you our analysis of these 5 different patterns and start with the granular one down in the lamellar bodies in the topmost stratum corneum cells. And this looks like this. And it's very high resolution images I will show you. The pixel size here is 1.8 angstrom, that is 0.18 nanometer. This pattern is replaced gradually by a lamellar pattern, the blue one, the 50 to 55 nanometer periodicity pattern that is lamellar, and it looks like this. And when we come up to the first stratum corneum extracellular space between first and second cell, it is replaced by the fine lamellar pattern, which looks like this. You see the periodicity is much smaller here. It's only 20 to 25 angstrom. And this pattern is in turn in the next extracellular space replaced by this pattern, which is an asymmetric fine lamellar pattern with a periodicity of 55 to 60 angstrom, and which finally then is replaced by the yellow pattern above the third extracellular space, which is the major barrier structure, which is also asymmetric, and it has a periodicity of 110 to 120 angstrom. So now the question is, what is the underlying structure of every one of these patterns? And this we have used our methodology, like I described before, by building molecular dynamics models and simulating what they would look like in our cryo-electron microscope. And for every of these analyses that I will show you, there are several publications that you can find in the overview paper that is referred to on the first slide. So this granular pattern, it is in reality a highly folded bilayer like this that exists in a lot of water. So it's swimming in a lot of water, this highly folded bilayer. And it's composed of not ceramides, but glycosylceramides, which is a ceramide with a sugar group attached to the head. And then, of course, the free fatty acid and cholesterols in a 1:1:1 molar ratio. And then the next pattern, the blue one, the lamellar pattern in topmost lamellar bodies with the 50 to 55 nanometer periodicity, it looks like this on the molecular level. And essentially, what you should retain is that you don't have any glycosylceramides anymore. Now it's ceramides. And it's a mixture of two conformations of ceramides. We have both ceramides in this hairpin conformation and ceramides in the splayed conformation when they're stretched out. And another feature is that all molecules, all the ceramides, the free fatty acid and cholesterol, are randomly oriented in both directions. So there is all molecules are in all directions in the lamellar plane. Then the third structure, that is the fine lamellar 20 to 25 nanometer pattern. The essential difference here is that you don't have any hairpin ceramides anymore. There are only splayed ceramides. And you also have an interdigitation of the lipid chains. That is, there is an overlapping of lipid chains. And this is what gives the fine lamellar appearance compared to the previous pattern. Still, you have a random orientation of all the lipids. Then is the fourth pattern, that is the asymmetric fine lamellar pattern with 55 to 60 nanometers periodicity. And the essential difference here is that the molecules have sorted. Free fatty acids and cholesterol, they don't like each other. So they have separated into different bands in this structure. You can see in this band, there is only cholesterol, no free fatty acids. And in this band, there are only free fatty acids and almost no cholesterol. And the same is true for the ceramides. The ceramides long chain and the short chain, they don't like each other. That's one of the reasons why they try to stretch out when they can. So they also separate into different layers. So in this layer, you have only short chain parts of ceramides. And in this layer, you have only long chain parts of ceramides. And then still an interdigitation like here and before. And then in the last one, the only difference is that the chains are not interdigitated anymore, but they separate completely with no interdigitation. Otherwise, the same structure with separated free fatty acid and cholesterol and separated ceramide long and short chains. So now is the question, how does these transform into each other? That is the actual process of skin barrier formation. And for that, we have also several publication for each steps, is the arrows here, so that you can find references to in the review article that's referenced on the first slide. So if we start here and take out, for visibility reasons, a part of this, the granular structure here, it's like this. You see the essential structure here is a bilayer of glycosyl ceramides. And you should imagine also, for visibility reasons, I don't mark here the free fatty acids and cholesterol, but for every long chain, there is one free fatty acid associated with it. And for every cholesterol, for every short chain, there is one cholesterol associated with it. So what happens here at the interface between the stratum granulosum and the stratum corneum in the extracellular space is that there is an enzyme called glucosylceramidase, which cuts glucose groups or sugar groups from the glycosyl ceramides. They cut the heads of them and form ceramides instead. And this means that the molecules become much less hydrophilic, because a glycosyl ceramide is the sugar groups that binds the water. Now it doesn't have the sugar group anymore, and it doesn't like water as much anymore. Ceramide binds only half a water molecule per lipid, while a glycosyl ceramide binds about 16 water molecules per lipid. So the system becomes unstable when the glucose groups, the sugar groups, are cut. So it collapses into a multilamellar system. You can see here that the bilayers become closer together. And at the same location, we also know that the skin is dehydrated, because in the topmost stratum granulosum, the water concentration is, like in all living cells, about 70%, while in the horny layer, it's about 10 to 30%. And we know also from cryo-electron microscopy that the extracellular space is more or less completely dehydrated right at this spot where this happens. So the next step is dehydration. And the effect is that the bilayers come closer together. You see, it gets less and less water. And at the final point, there is no water between the layers anymore. And then the head groups of the opposing bilayers start to touch each other. And this means when it's no water anymore associated with the ceramide head groups, they can now stretch out into their preferred conformation, because it's the water that forces ceramides and also glycoside ceramides into the hairpin conformation. When they can, if there is no water, they prefer to stretch out so that the long chains become separated from the short chains. So what happens is that there is a molecular stretch out at this point. This is where you see the blue pattern. And this progresses continually until all of the ceramides have stretched out into the splayed, elongated conformation. Once that is done, they start to reorganize. Now the free fatty acid and cholesterol, they want to separate into different layers. There is no water anymore that forces them together. The same is true for the splayed ceramides. You see here, they are mixed, the short and long parts of ceramides. They want to avoid that, so they start sliding along the length axis. And they slide more and more until the long parts find other long part ceramides, and the short parts of ceramides find other short parts. So it becomes like this: only long chains in one layer and only short chains in another layer. And you should imagine also the free fatty acids associated here with the long chains and the cholesterol associated here with the short chains. And when that is done, finally, they separate out, so there is no overlapping anymore. This is the conformation that has the lowest energy, the most stable packing, and therefore the best barrier capabilities. And this is the final, the yellow pattern, the pattern with 110 to 120 nanometer periodicity. So basically, as a sum up, in the stratum corneum extracellular space, we have the barrier structure that has this basic organization, and which the models in fits extremely well with the cryo-electron microscopy data. But not only that, also, if we look at how they stain the model, they stain in an optimal way, the same as what you see in conventional electron microscopy. Here you have the skin's barrier structure, that is the same structure as in the cryo images, but stain. The contrast is received from the stain in conventional electron microscopy. And when we stain this model, we get the same pattern as in conventional electron microscopy. So basically, the skin's barrier structure is like this. You see again how it's a bilayer structure with stretch out ceramides, and where the long chains are in a separate layer and the short chains in another layer, so they avoid each other. Also, the free fatty acids are in the long chain layer, while the cholesterol are in the short chain layer, so they also can avoid each other. And then we use this model for now our work is for calculating and studying how it's deranged in different skin diseases, like for example atopic dermatitis. And I would like to show you what the actual, in reality, the molecular dynamics model, what it looks like, because these are dynamic models. So I will show you here in full screen how it moves. And I've understood that you don't see it as smooth as I do because of the internet connection, but I hope you can see something at least, that these molecules, these lipid molecules, the ceramides, which are the green ones, the free fatty acids, which are the orange ones, and the cholesterol here, cholesterol carbons are gray instead of yellow, as I showed before. And then you see the acylceramides, these are the blue molecules here. And we, in molecular dynamics simulation, we simulate the function of this barrier structure. And also now we try to understand how the barrier structure is functionally, both structurally and functionally, deranging, for example, atopic dermatitis. And another application is that we use this model for calculating how drugs permeate the skin. And there I can show another image of drug calculations from the skin, how to optimize drugs for dermatological treatment. And this is actually a testosterone molecule that is penetrating the skin's barrier structure. And then we can simulate different ways to formulate it in different cream, for example, application, and see how the cream molecules, penetration enhancers, interact with the barrier structure and how it helps the drug molecule to get through in an optimal way. Okay, so that was my presentation.
Thank you very much. Thank you very much, Lars. It was very clear, and thanks for sharing these fascinating models to study the skin barrier. And we have time for some questions. So there is a first question we received. So thank you for such a clear and detailed description of your amazing work. Is it possible to look within the corneocytes themselves to gain information about their contents?
Absolutely. We have spent many years looking inside the stratum corneum cells at very high resolution, at actually even higher resolution than with the images I showed you. We have our data is with a pixel size of only 1 angstrom when it comes to the keratin. So our absolutely best data are actually from keratin. We have not yet published that. And the reason is that the keratin molecule is enormous. And we, for the structure determination, we have to calculate the complete keratin dimers and then the whole filaments. And this is an enormous computational work that is ongoing. And that's why we haven't published it yet, but we have very nice data on keratin inside living corneocytes.
Absolutely amazing. Do you have any idea about how this looks like in other vertebrates, in animals? Is the human stratum corneum very special?
We have not studied other animals. We have just studied human stratum corneum, but we know from conventional electron microscopy. And there lots of different stratum corneum have been studied. Frogs, for example, birds, and of course mice and guinea pigs. And there it seems that it's the same basic pattern of the barrier structure. Is this so-called broad, narrow, broad electron density staining pattern, which is famous which is known both in pig and mice and human. But it seems, even if there's much less data, probably also in bird and in frog. So it seems like this basic barrier structure is actually preserved and more or less the same, even if the composition in lipids can vary a little bit between species. It seems the basic structure, according to conventional electron microscopy, that it is the same, but we have not studied it with cryo-electron microscopy.
That's good.
Thank you very much.
Thank you.
No, thank you very much. There is another question, a very specific question from Leopold Eckhart. So thanks for these great presentations. What is the evidence for the conversion of the folded lipid bilayer into stacks of flat bilayers? Is this process temperature dependent? How is it affected by lipids?
Yes. If you look in this overview paper, there is one reference to Wenberg et al., where we, with molecular dynamic simulations, has simulated this transformation between the highly folded and the lamellar. And what is done there is exactly what is happening in the skin, that in the computer, the sugar groups are cleaved off. And the system, then we just follow the system when the sugar groups are cleaved off the cerebrocytes. And the system is gradually dehydrated, like is happening in skin. And then completely spontaneously, without us interaction in any way, the folded membrane collapses into a multilamellar bilayer, exactly the way I showed you here. So just look, and there are videos also available in this. So Wenberg et al. In the overview paper, the reference list.
I think we can take, there is one last question, maybe with a brief answer. So the question is from Pierre Sohier. Thank you for your great presentation. I assume you need a lot of prior knowledge on the chemical composition of the structure you are studying to be able to build accurate models. So how sensitive would be those models to incomplete knowledge about the structure or the disease? Would this modeling technique extend to other skin layers, maybe less pre-existing knowledge?
Of course, it all depends on the knowledge. You can do an infinite number of guesses, but the more knowledge you have, the fewer the guesses you need to make. So the closer you can come from start will facilitate it. In this case, we had a lot of knowledge. It's a fairly simple system. It's very well known, the composition. It's a fairly simple composition, and we know the relative amounts of the difference. So, and already then, we had to do several thousand guesses until arriving. And actually, the method is extremely sensitive, especially thanks to this focus trick that we use, because these are interference phenomenon that appear in the phase contrast in the microscope. And these are extremely sensitive. So even if we change the model I showed you, only a few atoms or a few percent in composition, we will get visible differences. They will not fit exactly as well anymore. So yes, it's true, you need knowledge, otherwise you will spend the rest of your life guessing. So therefore, biochemical knowledge, for example, the more the better.
Thank you very much again, Lars, for these great presentations, very clear. And so we'll receive some other questions, but we do not have time, so we will send you all these other questions, and your talk will be available on the ESDR channel on YouTube. So it's time to conclude this session, and thanks everyone for your attendance to this seminar. So the next seminar will be June 1st, freshly baked, and information will come soon. The next one will be June 15 with two speakers for the sweet and sour webinar. So biomarkers in inflammatory skin disease with Catherine Smith and Kilian Elrich. The deadline reminder for the deadline of the submission for abstract for the ESDR meeting, May 27. And then there is also the Global Health Awards, and the closing date is May 20. Thank you very much again.
Thank you.
Bye-bye.
Bye-bye.